Hello, everyone, and welcome to part 3 of the lecture on Chapter 10, Controlling Microbial Growth in the Body. Where we left off with the end of part 2, whoops, I should have done this. We were looking at ways of disrupting the cytoplasmic membrane and cell membrane, plasma membrane, all means the same thing. Oh, there's one piece that I forgot to mention. When we were talking about this ergosterol, just like cholesterol, there's a piece of it. This is the sterol, the 4-ring structure of the sterol that is lipid soluble, and that's the part that hangs out here with the lipid tails. And then there's this more hydrophilic part that hangs out with the phosphate heads. And so that is why these sterol molecules line up the way they do in the cell membrane, and it's possible for the amphotericin B to help create a pore. So there's an opening in this cell membrane now. Okay, that was an aside. We're going to continue with different mechanisms of antimicrobial activity. This was the second one, and I'm going to put that collection of 4 back up here. These are the 4 mechanisms. We already hit inhibition of cell walls. That was things like the beta lactams and interfering with the binding of the NAM units, and then the fungal cell walls. There are some drugs that we don't talk about very much, but there's some drugs that work on the fungal cell walls. Most of the fungal drugs actually work against the plasma membrane. And with bacterial antibacterial drugs, mostly we inhibit the formation of the cell wall, and there's a little bit of drugs that inhibit the plasma membrane of bacteria. Now we're going to move on to inhibition of protein synthesis. In this case, what we were talking about is disrupting bacterial or prokaryotic protein synthesis. And then finally, we'll finish up this section of the lecture with inhibition of metabolic pathways. And I'm going to give you one main class of drugs that works like this, but there are others. Okay, so let's start with inhibition of protein synthesis. Let me remind you, to get a protein, to make a protein, you have to start with the information in DNA, do transcription to get that messenger RNA, and then that second step, going from transcription to, or going from the messenger RNA to making a functional protein, you have to do translation. And that is where most of these drugs act. This inhibition of protein synthesis is impacting translation. That's why we spent so much time on translation. So now we're going to inhibit this protein synthesis in bacteria. And how we do it, remember, we're talking about selective toxicity. That is the ability to target structures or functions in a particular organism that's different from the host organism. I'll remind you that prokaryotic ribosomes are so-called 70S ribosomes. They're made up of 2 ribosomal subunits. There's a small ribosomal subunit, in this case 30S, and a large ribosomal subunit, 50S. And the next thing I'm going to tell you about this S, I promise you, is completely FYI, but some of you are going to get stuck on the fact that 30 plus 50 do not add up to 70, and you are correct, they don't. This unit is not a measurement of like mass or anything like that. It's actually a measurement of time. And so the way that the S stands for Svedberg. I'd look down to see how what the word was, because I always forget it. Svedberg units are, it's how long does it take this thing to get to the bottom of a tube if I'm spinning it in a centrifuge and it's suspended in a sucrose gradient. It's very specific, but that's why these numbers don't add up. So when you take this whole ribosome, it takes this long to get to the bottom of the tube. If you break it apart into the small ribosomal subunit and the large ribosomal subunit, they take this long and this long, respectively. That's why the numbers don't match. Okay, at any rate, prokaryotic ribosomes are 70S. their structure is a little bit different from most of our eukaryotic ribosomes, are 80S. And again, these numbers aren't going to add up either, and for the same reason. Take the two subunits apart, the little one comes down this fast, and the larger one takes this long to get to the bottom. Okay. Some of the drugs that we use that target the 70S, some of you are already thinking to yourself, wait a minute, don't I have some mitochondria? And mitochondria are believed to have evolved from what once were free living prokaryotes. And yes, those ribosomes in our mitochondria are similar to these 70S ribosomes that you see in other bacteria. So yeah, some of these drugs affect our cells too, but they affect the bacterial cells more than they affect our cells. Okay, so then these drugs can selectively target translation, just different aspects of translation. Some examples of drug names that you may have heard of already: streptomycin, tetracycline, erythromycin. And on the next slide, we're going to see 6 different mechanisms that these drugs can use to inhibit protein synthesis, inhibit translation. Ta-da! And we're going to march through them one at a time. So over here, let's start with A. This one, remember, let me remind you, this red thing represents the messenger RNA. Then there's a large ribosomal subunit and a small ribosomal subunit. And these drugs, known as aminoglycosides, what they do is they change the shape in that small ribosomal subunit so that this messenger RNA doesn't lay exactly the way it's supposed to. And so remember the way translation worked, we need the transfer RNA molecules to come in and be carrying an amino acid on the other end. Now, what this depends on is matching a codon here on the messenger RNA with the anticodon on the transfer RNA. And if this thing is all wonky, then this matchup isn't going to be the way it's supposed to. In other words, the transfer RNA that comes in and binds here is the wrong one. And so we end up getting what would essentially be a missense mutation. You bring in the wrong amino acid. So if you do that, that protein is not going to be shaped right. And so it's not going to be able to do the job that it's supposed to do for the bacterium. Ha! Genius. All right, so a drug that works like this, streptomycin, is one that affects the shape of the small ribosomal subunit. Maybe a way to remember that, let me see if that is, maybe it's a way to remember it. Remember that we said streptococcus, the strepto means a strip. So streptomycin is impacting the strip of this messenger RNA. It's not nice and straight anymore. Okay, next one. This is a drug you've probably heard of, tetracycline. You may have taken it yourself. What it does is it lays across the messenger RNA in a way that does not allow the transfer RNA to bind here anymore. So it blocks the docking of that transfer RNA. This one was harder for me to remember. What I finally came up with for myself to remember is tetra means 4, and cyclin is circle or ring. So this is a 4-ring structure. And if you look at a 4-ring structure, there's basically 3 little slots between the rings. And that's finally what I had to use to remember. Okay, that's the 3 little slots of the codon. And so this thing's going to block the codon and not let a transfer RNA molecule meet here. And so now this translation process has been interrupted in the bacteria. All right, the next mechanism, this is my absolute favorite because it's so clever, chloramphenicol medications. The way these work, they let all the translation happen in some sense. The small ribosomal subunit binds with the messenger RNA, and then the large ribosomal subunit comes in, and then remember AUG, that initiation, and then you get the, what's it called, the methionine as the first amino acid that comes in. But here's how clever it is. Chloramphenicol essentially makes it impossible for this ribosome to catalyze the reaction it's supposed to catalyze between the amino acids to get that peptide bond. And so this whole thing keeps marching along, yeah, bring me the next amino acid, great. And then as it brings them in, it releases them because there's no peptide bond formed. And so, yeah, you keep doing your thing, buddy, but you're never going to have a functional protein because you're never making oh my God, the bonds between the amino acids. So, so clever. All right. That's the third one. Now let's move on to the fourth one. The lincosamides or macrolides, they bind to the large ribosomal subunit, and basically they stop the movement of this ribosome. They stop the movement along the messenger RNA. And I'll give you a trick to remember this. Erythromycin is one of the drugs that can do this trick. Remember from erythrocyte, erythro means red. So erythro, red like a stop sign. This stops the movement of the ribosomes and in that way stops translation. And once again, it's stopping translation on the bacterial ribosome. So the bacteria can't make the proteins that they need to make. Okay, next one. This is antisense nucleic acids. So if you know what the code is in the messenger RNA, and we know what a lot of the proteins are that these bacteria are trying to make, then if you synthesize the complementary strand of RNA, you get something that's going to bind to the messenger RNA and essentially make it impossible for the small ribosomal subunit or the large ribosomal subunit to bind there. And again, translation can't happen if you can't get the ribosomes and the messenger RNA together. Okay, and then finally, the last one, oxazolidinone. Oh my gosh, oxazolidinone. This is really quite clever. This binds to the one of the sites. Where's my cursor? One of the sites of the ribosomal subunits and does not allow the large ribosomal subunit to join. So the first bit of translation starts, the small ribosomal subunit, and that first transfer RNA that binds to the AUG, that all starts. But here's the oxazolidinone, and large ribosomal subunit cannot bind. Why does this matter? Because this is currently one of the drugs of last resort to treat certain bacteria that have evolved mechanisms to defend themselves against vancomycin and methicillin. I know that you've heard of the methicillin-resistant Staphylococcus aureus. This is MRSA, MRSA, methicillin-resistant Staphylococcus aureus. The Staphylococcus aureus is bacteria that lives on most of us. And we have it on our skin, and it, usually not a big deal, but there are some strains of Staphylococcus aureus that can no longer be fought using methicillin. And some of them also cannot be fought using vancomycin. And so oxazolidinone is one of the last antibiotics that can still be used to fight some of those. Unfortunately, and perhaps not surprisingly, there have been cases of bacteria that have shown to be resistant even to this antibiotic. But it's the best we've got remaining to fight off MRSA. All right, so that is 6 different mechanisms of interfering with protein synthesis in bacteria. Generally, when a drug is prescribed, the idea is that the medication is going to fight this particular bacterium or this particular strain of bacteria as focused as possible. And we'll get to that a little bit later in this chapter. But if multiple antibiotics are going to be prescribed, you wouldn't pick multiple antibiotics from the same category. You pick like one if it's going to block the mechanism, or it's going to block protein synthesis this way. You don't need to block protein synthesis in another way. You're already blocking protein synthesis. Go pick a different drug that does something else. That would be more effective. That's why knowing all of these different mechanisms is helpful. That it's all, what they have in common is that they're all blocking protein synthesis. All right, and now we will move on to the fourth and final way that antimicrobial drugs can work. And this is a kind of a catch-all category, inhibition of some other series of reactions or reaction in the microbe, something that's going to be different between the microbe and us, the human cells. So you can do that if you can identify a pathway that's going to be different between the pathogen and the host, which is not always the case. But okay, so some of the early attempts, heavy metals such as arsenic can inactivate enzymes. Unfortunately, they can inactivate enzymes in both the bacteria and in our cells. So nowadays they're not used so much, but you may or may not have known this. I know I mentioned it in the lecture earlier, that arsenic used to be used as the treatment for syphilis. It had to be titrated very carefully to fight off the infection without killing the host. But there are other drugs that aren't quite so dangerous for our cells that still have an effect on the cells of the bacterium. The ones we're going to focus on are the sulfonamide drugs. These were discovered after penicillin was discovered, but became a kind of a functional drug that could be used and prescribed and used more widely before we kind of got a handle on how to do penicillin. So the so-called sulfa drugs. And I'll explain it, but basically it boils down to it makes it hard for the bacteria to make more DNA and RNA. And as you know, you can't make offspring cells without copying the DNA and replicating that information to pass it along. And so this sulfonamide drugs interfere with the ability of the cells to reproduce. And they interfere with the bacterial cells more than they interfere with our cells. All right, I know there's a lot on the slide, but it's easier to just have it all up at once. So the sulfonamides. What this all boils down to is that there is the end product is making DNA and RNA. And there's a molecule that's required way early in the pathway over here that we can interfere with this first step of this pathway. Okay. So the molecule we're interested in not allowing to be processed is something called PABA. That's how I'm going to refer to it, as PABA, for those of you who love detail. Para-aminobenzoic acid. That's the last time I'm going to say that. Let's call it PABA. It's shaped like this up here. And notice, I'm not going to test you on details, but I do want you to notice the detail of this. Everything in the pink boxes. We have an amino group that's a nitrogen and a couple of hydrogens. And then here's a benzene ring. That's a 6-carbon ring. And then there's some other parts to this molecule over here, but the important part for our purposes is this, the amino group and the benzene ring. Notice over here, we have an amino group and a benzene ring, and then there are some different parts of the molecule over here, sulfur, another amino group, etc. Over here, a third version of this, but what's identical is the amino group and the benzene ring, and then there's some other part of the molecule, and finally, once again, amino group, benzene ring, and some other part of the molecule. And why this all matters is that this part of PABA fits into the active site of a particular enzyme. And the processing or the job that this enzyme does on PABA will only work if the amino uh, group and the benzene ring can fit here in this active site. And then you're supposed to process it, and then, okay, okay, let's go into it. Dihydrofolic acid. You may recognize the folic acid part of this. Folic acid is something that we use also. You may know it as vitamin B9 or folate. This is also necessary in us for DNA and RNA synthesis, but in us, we don't even do this step. We bring the folic acid in our diets. We have to get that in. It's one of the necessary vitamins. But bacteria have this other mechanism that they can use to make folic acid for themselves. And the drugs that can fit here in this active site will actively interfere with this process. And so any of these, okay, so here's the PABA with its little carbon and double bonded oxygen and hydroxyl group. That's PABA. In contrast, here's something that's fitting into the active site of the enzyme with the amino group and the benzene ring. But wait, that's a sulfur. There's a couple of oxygens. That's a sulfur. There's a couple of oxygens. This is sulfonamide. That's going to bind in that same active site. It's not going to become dihydrofolic acid. It won't, because it's not PABA to start with. But PABA can't get in here because this drug is blocking the active site. Yay for fighting off bacteria. All right, so that's how the sulfa drugs work. They fit into the active site of this enzyme, which means that enzyme can't catalyze the reaction that it's supposed to catalyze. And so, although it's interfering way over here in the pathway, the end result is the bacterium cannot make enough DNA and RNA to copy itself. So clever. All right, and that is going to be the end of this part of the lecture. See you over in part 4.